DNA and RNA are the two molecules that store and transmit genetic information in living things. Understanding the structure and function of each and being able to explain their similarities and differences is essential to your success as an AP Bio student. In what follows, we'll see how both DNA and RNA are nucleic acid polymers, but that the monomers that make them up, which are called nucleotides, are different. We'll also see how the fact that DNA is a double helix while RNA is single-stranded explains the different function that these molecules play in living things. Along the way, we'll talk about some amazing issues like the genes of viruses and the origin of life. Our goal to make sure that you know exactly what you need to know in order to ace your next test and to crush it on the AP Bio exam. Hey, I'm Mr. W. I'm a singing retired AP Bio teacher, and my mission is to help you succeed in biology. In addition to this video, which is based on teaching materials that I spent an entire career developing, I've got two other things that are really going to help you succeed in your AP Biology course. First of these is my website, learn-biology.com, where there's an entire interactive AP Bio curriculum. And in addition to that, I have an app that's called Biomania that has quizzes, flashcards, interactive free response questions, and games that will help you learn more biology than you ever imagined possible. I'll tell you more about those later on in the video. So right now, let's do it. Let's learn about DNA and RNA, their similarities and their differences. DNA and RNA are both classified as nucleic acids, and that means that they're both polymers of nucleotides. Nucleotides are the monomers that make up the nucleic acids, but the nucleotide monomers are slightly different. Nucleotides consist of three subparts. At the center is a 5-carbon sugar. That 5-carbon sugar is called deoxyribose in DNA. It's called ribose in RNA. In addition, there's a phosphate group, and what you're looking at here is the ionized form of that phosphate group, and that means that there was once a hydrogen over here and over here, same thing on this side, but what happened is that when DNA and RNA are in water, those go into the solution and they make the solution acidic. That's why we have deoxyribonucleic acid and ribonucleic acid for RNA. Finally, in addition, there's a nitrogenous base. Nitrogenous means that there's a lot of nitrogen. And in addition, there's an amino group shown at number 4 over here. The amino group is the basic functional group, and that makes these nitrogenous bases. Now, you'll see that there are these little red numbers. Biochemists have a system for numbering the carbons in a sugar or any molecule for that matter. You don't need to know how to apply that, but you do need to know that, for example, this is the number 1 carbon. Now, how is it a carbon? Well, in this kind of structural formula, wherever there's an angle vertex without any specific letter, that means that you assume that it's a carbon, because carbon is the basic molecule that we find in living things. So to talk more about the structure, note that the number 1 carbon is what's linked to the nitrogenous base. The number 5 carbon is what's linked to the phosphate group. And notice this difference over here. The number 2 carbon in deoxyribose is linked to a hydrogen atom. The number 2 carbon in ribose in RNA is linked to a hydroxyl group with an oxygen. Now, notice something cool. This is ribonucleic acid, RNA. Well, if you take out an oxygen, you've deoxygenated ribose. And that's why this is called deoxyribose. So now you understand why RNA is called RNA, DNA, deoxyribonucleic acid. Here are the 4 DNA nucleotides. Notice that every nucleotide has this 5-carbon sugar, deoxyribose, which is linked to a phosphate, but that the nitrogenous base can differ. In some courses, you might need to know the difference between the nitrogenous bases that have 2 nitrogen rings, such as adenine and guanine, and those which have 1 ring, such as thymine and cytosine. These are called purines. These are called pyrimidines. The way that I've always taught my students to remember that is that purine has 2 syllables and therefore 2 nitrogen rings. That doesn't really work for pyrimidine, but there's nothing with 3 rings, so just remember 2 syllables, 2 nitrogen rings. You'll often see these nitrogenous bases referred to by just the first letter of their name. So adenine is referred to as A, thymine T, guanine G, cytosine C. Here are the 4 bases that make up RNA, and you'll notice that there's 1 substitution. So we continue to have adenine and guanine as the 2 purine nitrogenous bases, but in RNA, uracil substitutes for thymine. So we have A, U, G, and C as the 4 bases that make up RNA. So we've introduced the monomers, the nucleotides that make up DNA and RNA. And now what we're going to do is see how they form polymers. What we see is the same in both RNA and DNA. Here we're looking at RNA, and you can tell because we have uracil over here. Basically, the way that the nucleotides are chained together, and they're chained together by enzymes, is that the sugar of one nucleotide connects to the phosphate of the next. Sugar over here connected to the phosphate of the next. And the numbering helps you see how this is organized. So the 3 carbon of one nucleotide connects to the phosphate of the next nucleotide, which is connected to the 5 of that very same nucleotide. This whole thing about 3 and 5 becomes particularly important when we talk about the synthesis of these nucleic acids. So like, for example, here you can see the process of DNA replication. And what happens during DNA replication is that an enzyme reads one of the strands of DNA and then uses the information to insert a new nucleotide. That new nucleotide is only inserted at the 3' end where that number 3 carbon is. That leads to a lot of complication during DNA replication, and I have a song about that, a DNA replication rap that you can watch, and I'll put together a lecture about that topic as well. Now, because of this ongoing sugar, phosphate, sugar, phosphate, sugar, phosphate, all the nucleic acids have a sugar-phosphate backbone. That's a structural part of these molecules. The information is actually held in the sequence of bases. And these molecules can be tremendously long. RNAs can be thousands of nucleotides long, and DNA in particular can be millions or even hundreds of millions of nucleotides in length. Now, the reason why these molecules, both DNA and RNA, are able to store information is because the bases can bond with one another, and that's because they have complementary shape. So there are base pairing rules for both DNA and RNA, and here's what they are. In DNA, thymine always bonds with adenine, and guanine always bonds with cytosine. Now you might be wondering, why is the adenine upside down relative to the thymine? Well, it's because they only fit together when they're oriented in exactly that way. So it's this kind of orientation, one upside down relative to the other. In addition to the fact that the bases are complementary, that complementarity enables those bases to get close to one another where they'll form hydrogen bonds. So here you can see guanine binding with cytosine, and here you see adenine binding with thymine. And these dotted lines over here indicate a hydrogen bond, a relatively weak bond that connects these bases together. And those bonds are weak, but over the length of the entire molecule, they wind up providing an incredible cohesive force that holds DNA together. And as I said before, the bases can only pair if they're upside down relative to one another. So the way that you describe that is that the strands are anti-parallel. In other words, if one strand is running in the 5' to 3' direction, then the other strand will have to do that in the opposite way in order for the two to bind together. So this is anti-parallel. This would be parallel, or this would be parallel. So that's how these bases actually bond together. Okay, so The hydrogen bonds that form between DNA give it incredible stability and allow DNA molecules to be phenomenally long. How long? Well, this is a picture of the chromosomes that are found in a human cell, and they're colorized for effect. But the thing to note is that you've got 23 chromosomes, and the first chromosome is the longest of all of the chromosomes in the human body. That chromosome, which is found in every single cell of the human body, is 285 million base pairs. So, you start counting 1, 2, 3, 4. You don't stop until you've gotten to 285 million. It takes a lot of information to code out an organism, and that's what that's all about. RNA also can form base pairs, even though it's not a double helix, and the base pairing rules are A bonds with U and C bonds with G. Now, why is that important? Because RNA can form hydrogen bonds within different parts of a single strand, forming loops and curls and things like that. But the thing to note is that, like, for example, in this part of the molecule, of a molecule called a tRNA, you're having hydrogen bonds that are happening between this area over here and this area over here. Same with this and over this. In other words, wherever the strands are coming together, there are hydrogen bonds that are stabilizing that shape. This is another representation, more of a 3D representation, where you can see the bases a little bit more clearly, but you can just imagine that somewhere in there, adenine is bonding with uracil and thymine is bonding with cytosine. That enables RNA to assume incredibly complex shapes, much like in the same way that a protein can form complex shapes. And that makes RNA a much more versatile molecule than DNA is. RNA is suited to play many functions within a cell, including functions that are much like the enzymes that you've come to know as you've learned about proteins. DNA does one thing: it stores information. During a process that's called transcription, what we have is the synthesis of RNA from a DNA template, and in order for that to happen, DNA and RNA have to bind with one another. So there's very simple rules that now you need to add to your already growing knowledge about DNA base pairing, RNA base pairing. There's DNA RNA base pairing. And the way that it works is G bonds with C, T bonds with A, C and DNA bonds with G, and A and DNA will bind with U in RNA. So you can just remember those rules, and then you'll be able to understand the genetic code and take on other challenges that you'll experience in your biology course in just a little bit. So let's summarize. What we've established so far is that RNA and DNA are both nucleic acids. The monomers that make them up are slightly different. In RNA, we have ribose as the sugar. In DNA, we have deoxyribose, and we understand why deoxyribose has its name. We've also seen that the bases, the nitrogenous bases in RNA and DNA, are a little bit different. In RNA, we have cytosine, guanine, adenine, and uracil. In DNA, we have cytosine, guanine, adenine, and thymine. We've talked about how the structural component of these molecules is a sugar-phosphate backbone, and what we've also seen is that there's base pairing that occurs in DNA to a lesser extent in RNA. It's core to the structure of DNA, and it's sort of an elaboration on the structure in RNA. And the result is we have single-stranded RNA molecules, and we've got double-stranded DNA molecules. We have some really important information that lies ahead, but what I'd like you to do right now is do me a favor. I'd like you to like this video. Please subscribe to my channel, Science Music Videos, and above all, I love questions. And if you ask me a question that I don't have a video for, I'll make you a new video based on it. So leave me a comment, ask me a question, and I'd really appreciate that. I also want to take this minute to tell you about the AP Bio curriculum that I have on learn-biology.com. It's a full interactive AP Biology curriculum. It's got tutorials, flashcards, multiple choice quizzes, and it'll give you the support that you need to do a very difficult thing, ace your AP Biology class. Because if you're feeling like this course is hard, that's because it is. It's a tremendous amount of information. It comes to you in a really, really fast pace. And if you need additional support, learn-biology.com is where you'll find it. Like, for example, you can see that we have these flashcards that will enable you to really master the differences between DNA and RNA. In addition to that, there's multiple choice questions and there's even games. I also have an app, it's called Biomania. It has a smaller subset of the information that you'll find on learn-biology.com. It's got flashcards, it's got multiple choice, it's got practice FRQs, and it's got diagram-based games that will be fun and they'll help you learn, and they'll also lead you to an incredible level of mastery. So let's talk about what's coming up next. We're going to talk about where DNA and RNA are found in cells. We'll talk about the functional differences between DNA and RNA, and we'll talk about the role of RNA in viruses and in the origin of life. Let's talk about where DNA is found in prokaryotic cells. Those are primarily bacteria. There's another domain that are called archaea. So in those organisms, you find DNA in two places: in a bacterial chromosome, which is a loop of DNA that can be hundreds of thousands or even millions of nucleotides long. And in addition, you find DNA making up much smaller loops of DNA that are called plasmids. And plasmids are used for exchange of genetic information between bacteria. It's called horizontal gene transfer. I have a tutorial about that on learn-biology.com. Plasmids are also widely used in genetic engineering. And when you learn about genetic engineering, you'll see how plasmids are relatively easily engineered, so you can introduce new genes in bacteria to get them to produce traits that aren't originally part of their genome. Where's the RNA? The RNA is everywhere. So the RNA would be right here next to the chromosome as transcription happens, and it'll be out in the cytoplasm, involved in protein synthesis and in other processes that RNA is involved in. In eukaryotic cells, there's more segregation of where DNA is located compared to where RNA is located. DNA is located in the nucleus. DNA will get transcribed and made into RNA. RNA will leave the nucleus where it'll direct processes like protein synthesis. And you'll see RNA in several forms that I'm about to talk about. But key thing, DNA in the nucleus, RNA in the nucleus, and in the cytoplasm. Now, let's talk about some functional differences between DNA and RNA. As I've said, DNA is a relatively limited molecule. DNA stores genetic information, and that's all that it does. So DNA gets replicated during processes like mitosis and meiosis, and DNA also gets transcribed into RNA. But that's what happens to DNA, replication and transcription. RNA goes into the cytoplasm, curls up into complex forms sometimes, and can do many more complicated things. So the types of RNA to know about are messenger RNA, which brings a message about what a gene should be out to the cytoplasm, what protein should be constructed from a gene. That gets read by a ribosome. A ribosome is a little nano robot that can translate messenger RNA messages and make proteins, and ribosomes consist of catalytic RNA that can actually put proteins together, along with some protein. There's another molecule called transfer RNA that's also involved in protein synthesis. And finally, you have a whole variety of small RNAs that play various roles in gene regulation. Now, here's two last things that are both really cool. We've talked about how DNA is the genetic material, and that's true in every organism on this planet, but it's not necessarily true in viruses, which aren't really organisms, because viruses aren't independent living beings. There are some viruses that have RNA as their genetic material, and that includes viruses that you're undoubtedly familiar with. One of them is HIV, the virus which causes AIDS, acquired immune deficiency syndrome, and the other virus is SARS-CoV-2, which is the virus that causes COVID-19 and put us all out of school for at least a year. So RNA is the genetic material in certain viruses. It's never the genetic material in cells, but only in some viruses. Finally, RNA, because it can form all of these complex shapes, is thought to be the first genetic molecule. So in other words, at some point during the origin of life, genetic systems had to evolve. And the idea is that as life was evolving, there was spontaneous emergence of inorganic precursor molecules. Those molecules, through inorganic processes, not processes that happened in cells, were combined to form molecules like the nucleotides that make up RNA. Those combined to form polymers, and at some point systems of those polymers together formed systems that could replicate themselves. That is sometimes referred to as the RNA world. At some point, those molecules became associated with DNA and or became encapsulated within membranes. And then we have something very, very close to a cell. So when you learn about the origin of life, you'll see that RNA plays a paramount role in that process. There is so much biology to learn, and what I'd like you to do is click this link to learn about the next topic in AP Biology.